My name is Nili Ostrov. My day job is a postdoc with George Church. And I would like to tell you about our project to recode E. coli and how could that lead, or what is the process that we think of when we think of expanding recoding to other organisms beyond E. coli. And so virus infection is a problem, and virus resistant is definitely a desired feature for cells that you use for industry or for therapeutics. The most famous example maybe is Genzyme having to shut down their factory for making drugs for, I do not remember the name of the drug, but it is a rare disease that they had to shut down the factory because of a virus infection. So the fermenter had to be taken down and drugs could not be made. And so what is a possible solution for that? One thing you could do is recode. So what do I mean by recoding? We all know that when a virus infects a cell, it inserts its DNA into the host cell. And so what if the DNA that the virus what if the DNA that the virus injects into the host cell could not be properly translated? And how do we do that? So we have a host cell, in this case a wild type E. coli cell, and it has, does this work? It has 3 stop codons naturally. What if we took every place on the genome that UAG stop codon is used and change that to a different one? And so if we change UAG to UAA, for example, then when a virus infects a cell and uses the missing codon that we now eliminated, that virus cannot translate its own proteins. That would make the host cell virus resistant. And you'll ask, does that actually work? And so this is work from Farron Isaac's lab using an E. coli cell where all UAG was eliminated and changed for UAA. And you can see that different viruses, when you test whether they can infect the cell, 5 of these 7 cannot infect the cell. 2 of these can still infect the cell. And so those 5 actually use this UAG codon that is now missing in the host cell, so they cannot infect the E. coli cell. Those 2 still have other codons that they use, so they can still infect. And so in our lab now, after doing the first E. coli that is missing a single stop codon, it is quite virus resistant, but we want to enhance that. We want all viruses to not be able to infect E. coli. So what we do now is we try to recode more than one single stop codon. And so this is the current project in our lab that we are doing, which is recoding of 7 different codons in the E. coli genome. We are taking leucine, stop codon, serine, and arginine, and we're eliminating those codons from the E. coli genome by replacing them with different codons for the same amino acid. So I want to just clarify that we are not changing the proteins. We're changing one codon for a different codon, both of which code for the same amino acid. And so to do that, we divided the E. coli genome into pieces, and we de novo synthesized all of the E. coli genome with genes, protein coding genes, that don't have any of these 7 codons. And so just to point out the differences between those 2 recoded strains. If you look at the genome size, E. coli is about 4 megabases. And so if you want to remove one single stop codon, you would need to change 321 positions on the genome. If you wanted to change 7 codons, you would have to change 62,000 codon positions. And so having done those 2, our lab now has kind of the experience to say, what does it take to do this, and what does it take to do a bigger scale recoding? And this project was done by genome editing. That is, oligos were used to change 300 positions on an existing E. coli genome. This project was done on the computer. The E. coli genome on the computer was redesigned to have all 62 positions of the 7 codons replaced. So synthesis of this genome basically took a computerized, designed genome and made all these changes into it, and then, if you will, printed out, synthesized the DNA to make a very, very modified E. coli. And so this E. coli, we expect, is going to be a lot more virus resistant than this one. And thinking of making a human or a CHA cell or any other cell line that is used by industry, you would look at how this scales up to other organisms. And so here I'm just looking at what would it take to change one single stop codon in either of any of those organisms. And so for Cerevisia, you already know there is the yeast project 2, which is doing, among other things, recoding. And you can see that more and more codons are necessary to change that. And what I want to convey here is that it is not necessarily either editing or synthesis. Those are very complementary. We're using a lot of different methods. Even the synthesis of a whole E. coli genome was not done just by synthesis. We assemble the pieces in yeast. We use MAGE, which is genome editing, to troubleshoot and to change our genome as we build it. And so the fact that we're going up in genome size does not necessarily mean that for a larger recoding effort, we have to synthesize the entire genome. And just a quick example, if you just look at the numbers, it would be a lot less to edit a human genome with a single codon recoding than to make the entire genome by synthesis from scratch. And we know a lot of the pieces that we're making here are not necessarily being recoded. They're not protein coding regions. And so what I would like to convey here is that there are a lot of different technologies that we are using to do this effort of recoding. And DNA technologies for recoding are very much available for E. coli, and we're hoping to soon have the first radically recoded E. coli done. But for mammalian and human cells, all of these technologies, including synthesis, assembly, CRISPR, and recombinering, they are not easy to do in human cells yet. And delivery into host cells is not something that is easily being done in mammalian and human cells. And I would like to point out that safe cell lines, if we can make those, are not only going to be virus resistant, there's a lot of advantages to recoding a cell, such as the ability to biocontain them and the ability to add new chemistry by introducing non-standard amino acids using recoded codons. And with that, I'll take any questions.
Thank you. Thank you very much for the nice talk. Very interesting. As you are well aware of, not all synonymous codons are born equal, and some are fast codons, some are slow codons. And this has been implied into the folding of the protein, the co-translational folding of the protein. So I wonder if you have any data from your work that speaks to this question.
Yes, that's a very important point. So in the interest of time, I didn't get into the computational design of the E. coli genome, but a lot of the success that we're having right now with being able to do this radical recoding comes from a lot of background and de-risking and other parallel studies in our lab for how do you actually look at the sequences and how do you recode them based on ribosome pausing, mRNA folding, all those things that come into, that by the way we don't know as well for mammalian cells, that came into the design. And we have a very robust computational design. We only have 25 genes that had a problem recoding out of 3,000 genes in E. coli, which is, I think, very impressive and very surprising to us also, but it's a good surprise. And so you're absolutely right that folding, mRNA folding, and a lot of other promoter and initiation issues should get into the computational design. Yeah.
Since I have it, it's a very cool approach, but is there really such a thing as an absolutely safe cell? If you change the codons, how long will it take for the virus to learn to adjust?
Yes. So that, I think, is a very, very interesting aspect of this work. One side is what would happen to the codon usage, so how would a recoded cell behave, and the other side is what would happen to the co-evolution of a virus with E. coli. And once this strain is done in E. coli, it will be very interesting to see the interplay between, for example, lambda-phage and a recoded E. coli. And so we don't know. We don't know how fast or if a virus would develop the ability to infect, but that would be a very interesting question to look at in these cells. George.
I'm willing to stick my neck out here and say that viruses will not be able to adapt to this, even with very huge virus populations, at least in our next thousands of generations of humans. Math. So if the virus has 1,000 different positions in its genome, which are highly deleterious, it has to fix all of those 1,000 positions from a starting point which is not viable. So if we're slogging along, like we were talking about drug resistance, yes, then each incremental change can make it a little bit better. But if you talk about something broken, completely dead for 1,000 different reasons, you can't adapt it. And the reason why this is not coevolution in the classical sense is that the virus wasn't watching while the host was evolving. You did it without the virus being able to see what's going on, and that's not fair.
We're still going to try it when it's done. Thank you.